Good morning. I'm Anna Torgasheva from Institute of Cytology and Genetics from Russia. And first of all, I want to thank organizers for inviting me. I'm very glad to be here, and I will talk today about a very strange chromosome, which is we now know present in all germ cells of songbirds and absent from their somatic tissues. Birds is one of the most diverse and numerous taxon with more than 10,000 species. They become more and more popular object of modern biological research, and about half of them are songbirds. And despite all the close attention, until recently, songbirds managed to hide from us a very big piece of their genome. In my talk, I want to tell what we know and especially what we don't know about mysterious germline restricted chromosome of songbirds. First, just to put this into wider context, the GRC is a curious example of programmed chromatin elimination. Naturally, we think that all cells of the body contain the same genetic material. It is not always true. Many organisms get rid of part of their genomes different stages during development. It is rather widespread from unicellular ciliates to some vertebrates. However, the mechanisms of DNA recognition and elimination of stages when DNA is eliminating, of type and amount of eliminating DNA differ dramatically between the species. There are two main forms: when whole chromosome or their fragments are eliminated during the sex determination, and second, when DNA is eliminated in the precursors of somatic cells in early embryogenesis. Germline, in this case, preserves the whole genome, and it happens in the germline restricted chromosome of songbirds. This story started more than 20 years ago when Argentinian researchers Pigozzi and Solari found an unusual excessive chromosome in germ cells of Zebrafinch. 16 years later, they described similar chromosome in Bengalese finch. These species are separated about 10 million years of evolution. However, in both of them, GRCs had the same properties. What did they actually find? Let me remind you some features about bird genomes. Birds have a relatively small genome, in average a little over 1 gigabase, with low number of repeated sequences in comparison to mammals. And the karyotype of birds is pretty stable. Most bird species carry about 10 macro and about 30 microchromosome pairs. Micro means really tiny. It's rather tricky to get a nice metaphase place with all chromosomes lying apart and count them with certainty. However, you still can count microchromosomes and determine their morphology. And what Pigozzi and Solari actually found was unusual element in the spermatogonial mitotic cells, a big acrocentric microchromosome without pair in male germline. It was the only acrocentric chromosome in the set. It was absent here in the somatic tissues, in all somatic tissues they checked. They called it germline restricted chromosome, GRC. What happened to this chromosome in germline during meiosis? During meiosis, homologous chromosomes pair with each other and recombine. Synaptonemal complex polymerizes along homologous chromosomes and binds them, forming bivalents. There are lateral element of the synaptonemal complex and central element, which is complete at packet end. Then it gradually depolymerized and the homologs split apart and segregate. In both finch species, GRC was condensed during male meiotic prophase. It was shown using antibodies to the modified histone specific for transcriptional inactive chromatin. Closer to the end of the prophase, it moves to the nucleus periphery and then is ejected from the nucleus after meiotic divisions. It forms round dense bodies near the spermatids, and it is absent in sperm cells. So here's a packet-end spermatocyte of Bengalese finch. There are 40 bivalents. You can easily and accurately count them, even microbivalents, and there is one big acrocentric univalent. This dotted line, this only lateral element formed. So what happened to this chromosome in females? In females, there are usually 2 copies of GRC in packet of most birds. 2 GRCs behave like absolutely normal chromosome pair. They properly pair and recombine. So it is a 1 copy of GRC in males, which is expelled after meiosis, and 2 copies in females, who apparently transmit it to the next generations. So this intriguing discovery raised a bunch of questions. Is this chromosome a genomic parasite, or is it an essential part of the genome? Is it really just a curious exception and present in only 2 related species, or more widespread? 5 years ago, we worked on a project on meiotic recombination in birds, and we studied 2 swallow species, St. Martin and barn swallow. And in all sites of St. Martin, we saw this. Strange, big acrocentric univalent, additional to the basic chromosome set. It was not one of the unpaired sex chromosomes, so they're normal paired bivalents. Blue dots here are centromeres, green dots are recombination sites. The diploid number of St. Martin was known, and this chromosome was definitely extra. We suspected it was GRC. And in bone marrow metaphases of this chromosome was absent. And this chromosome looked and behaved in meiosis exactly like GRC of zebra and Bengalese finches. Most females carried 2 GRC copies, which properly pair and recombine in the ends. Most males had single GRC copy, which form univalent. What is notable here, anti-centromere antibodies that we used to mark centromeres usually marked chromatin of the unpaired GRC in males. After meiotic divisions in males, GRC also eliminated from nucleus. We also saw dense bodies near spermatids, and it was apparently absent in sperm. So we had a pretty much similar chromosome in the species diverged about 40 million years ago. We also found GRC in the sister species of the St. Martin, pale martin. And we didn't find any big acrocentric chromosome in packet cells of the barn swallow, which is diverged from St. and pale martins about 30 million years ago. So here comes a reasonable question. Do finches and martins have the same germ-like restricted chromosome? Did it appear in the common ancestor of the finches and the martins more than 40 million years ago, and then lost in barn swallow lineage, or did it evolve independently? So first, we need to ask, do finch and martin GRCs have common genetic content? We decided to assess it with chromosome-specific probes and cross-species hybridizations. And we used the unique feature of GRC, its elimination from spermatocyte. We microdissected round dense bodies lying near the spermatids and got GRC-specific whole chromosome hybridization probes for all 4 species. Hybridizations we used as obtained probes first showed that these bodies are indeed GRCs. The probe marketed here and at packet cells. The probe intensely lapped GRC of the same species. Besides GRC, there were weak signals at different chromosomes of the basic set. In pale martin, it was pure centromeric regions. In Zebrafinch, there was a bright signal at the short arm of chromosome 3. This indicates that GRC apparently contain repetitive sequences, and at least some of them are homologous to dispersed and clustered genomic repeats. In the interspecies hybridizations, signals on GRC were much less intense and did not cover the whole chromosome. Again, there were signals on basic chromosomes, different for different GRC probes. It tells us that there is something in common between zebra and Bengalese finch GRCs. They probably share some repeated sequences, but obviously, mostly GRC contain different genetic material. In the cross-species hybridizations between martin and finches, labeling on GRCs was even less intense. We can suggest that the GRC evolves very fast, which is expected because the selection pressure is decreased much for GRC. since it is excluded from most cells of the body and it doesn't recombine along most length. So microdissection probe gave us a lot of information to think, but it did not answer the question about GRC origin, because what GRC of different species share might be just genomic repeats. So we obviously needed more birds. And we found no signs of GRC in published data of species outside songbirds. Then we checked available songbirds and we found big acrocentric extra chromosome in bullfinch, grey-tit, and warbler. It drove us to the idea of GRC origin in the common ancestor of the songbirds, but how is another question? Why carry such a big potentially parasitic chromosome in all germ cells? Apparently, some species lost it. There are no big acrocentric chromosome in pit, flycatcher, common starling, white wagtail, and barn swallow. It means it is not necessary. So we asked what is special about birds with and without GRC. It turned out that all our birds without GRC were females. In females, GRC form fine bivalents. So the only way to notice them is comparison between meiotic and somatic karyotypes. It is easier when GRC is large. But what if GRC is a microchromosome? We could have missed it, since for most birds, the exact diploid number is not determined. We understood we need a barn swallow male. And indeed, in barn swallow spermatocytes, we saw this. A tiny acrocentric dot-like univalent labeled with anticentromere antibodies. The number of properly paired bivalents in females was 41, in males 40, plus this, and it was painted with GRC probe of St. Martin, along with several other microchromosomes. So in females, it's just one of the microchromosome. And then we found GRCs of different sizes in more than 10 more species. In goldfinch, pine bunting, rook, golden finch, skylark, Eurasian siskin, and the size of this chromosome differs much. So altogether, we found GRC in all studied 16 songbird species, and we found no evidence of its presence in all studied non-songbird species. It indicates that apparently GRC originated in the common ancestor of the songbirds, and indeed evolves very fast. It differs drastically in size, morphology, and genetic content. It apparently contains multiple copies of regions from basic genome. To make an insight of what these regions are, we sequenced our microdissection libraries. We aligned GRC reads to Zebrafinch genome, and found it's really true. Different parts of genome is multiply repeated in different GRCs. Some of them overlapped with genes. Here's a list of regions in Zebrafinch genome disproportionately densely covered with reads of GRC. Unfortunately, our data did not allow us to tell if there are any full-length genes, and the question was, does GRC contain anything functional, or it just collected debris traveled from basic chromosomes? And the answer came very soon. In parallel with our study, group of Alexander Su from Uppsala University sequenced GRC in Zebrafinch. They used complementary approach of comparative phylogenomics and came to the exactly same conclusion, that GRC is apparently originated in the common ancestor of songbirds. Also, using a transcriptomic and proteomic analysis, they showed that GRC actually contains at least more than 100 genes paralogous to the basic genome. Some of them are transcribed and some of them even translated. Most of these genes are involved in female gonad development. So what has changed in the 20 years since the first discovery of the GRC in Zebrafinch? We know it is widespread among songbirds and evolves very fast. We know it contains copies of regions from basic chromosomes, but we don't know if GRC contains something unique, not present in the basic genome. We don't know if GRC of different species share something functional. Probably if this chromosome deals with female gonad development, and it is logical because of its behavior, and it is confirmed by the sequencing of Zebrafinch. However, there are no data from any other species yet. There is also not clear how GRC is originated. The available data do not answer this question. However, we can suggest that GRC might have occurred as a microchromosome, potentially due to the non-disjunction. If it carried the genes necessary for the development, it became an essential part of the genome. And then, independently in different lineages, it might collect and amplify different parts of the basic genome. It is possible due to suppressed recombination on most length of GRC and due to the reduced selection pressure, since it is eliminated from most cells of the organism. The unusual behavior of this chromosome also raises a question about its inheritance. Since it is eliminated from males and since all birds carry this chromosome, they should receive a copy from the mother. But why females carry 2 copies of GRC and males carry 1? Actually, it is not always true. While most females in zebra and Bengalese finches carry 2 GRC copies, some females carry 1. The same we saw in St. Martin females. About 15% of females had a single GRC in all packet cells, which appeared as univalent. Most females had 2 copies. Also, most males had 1 copy of GRC, but unlike in zebra and Bengalese finches, some Martin males were mosaic for GRC copy number. In the mosaic, from 5 to 60% of cells carried 2 GRC copies. They usually looked like this, with partially paired ends, mostly unpaired. So it indicates that there are apparently 2 ways of GRC elimination, one during spermatogonial divisions and second after meiotic divisions. But what was even more surprising, we even saw cells with 3 GRC copies in males. One as unpaired univalent here, and the one as normally paired bivalent. What could potentially explain this very strange finding? There is a recent preprint where authors revealed that GRC in Zebrafinch sometimes can be transmitted via males. They showed it cytogenetically, they showed it by sequencing ejaculates and also by reconstructing the inheritance of GRC in captive breed population of hybrids between 2 zebrafinch subspecies. However, we did not see mosaic males in zebrafinch as well as the GRC in sperm of swallows, probably just because it is rare. But what should occur regularly is getting GRC to the egg after meiotic divisions in females, even in case of 1 copy. It is very difficult to analyze these stages in birds, but what we suggest that the observed picture of the inheritance might occur due to the meiotic drive in the first meiotic division. All zygotes, in this case, receive either 1 or 2 copies of GRC. These 1 or 2 copies stay in the oocyte after the first meiotic division, and then in M2, sister chromosomes segregate, producing an egg either with 1 or 2 copies. Most males in this scenario receive 2 copies of GRC, and they successively lost them during spermatogenesis. Of course, this scenario is our speculation, and hopefully future researchers will prove or disprove it. There are really a whole bunch of questions on biology of this chromosome. It is still unclear what is the mechanism and stage of GRC elimination. And maybe the most intriguing question is Does GRC have anything to do with the extraordinary diversity of songbird species? Could it play a role in the reproductive isolation and facilitate the speciation in birds? It is highly speculative, but tempting to think that GRC may serve as a spare corner for variation, especially there are not enough room in the basic genome. So I'm very grateful to people who made this work possible, and I will be happy to answer your question. Thank you.